All right, I think we will go ahead and get started. Delighted to welcome everybody to this webinar focused on funding related to HIV research going forward. And we're delighted to have a person who probably knows more than almost anyone about this topic, and that is Carl Dieffenbach. As many of you will know from having registered for this call, Carl is the director of the Division of AIDS of NIAID, the National Institute of Allergy and Infectious Diseases, which is part of the National Institutes of Health. For those of you outside of the U.S., that is an alphabet soup of acronyms, and frankly, it is for those of us in the United States as well. I think the important thing, though, to know in advance of this call is if you think about HIV research generally, I think everybody's quite well aware that the NIH, and particularly with NIAID as the lead institute in terms of HIV funding, is involved across the widest span of HIV research, from basic science to clinical research, product development, implementation science, across the board. And much of that work happens across many institutes at the NIH, but as I said, the lion's share of the funding, the largest funding for HIV research sits within the Division of AIDS at NIAID. And delighted to have Carl here, who is just shy of a decade of running the Division of AIDS at NIAID, and has really spent that decade leading the division and involved in pretty much every major research endeavor you can imagine. The NIH itself accounts for over 70% of HIV research dollars, whether that's in basic science or clinical research, whether for treatment, prevention, or cure. Carl, for the last decade, has led the division, which has a research portfolio of over $1 billion a year and over 150 employees working in the division. And one can't really think of many research findings in the last decade, whether that is HPTN 052, whether that is RV144, whether that is the VOICE study, whether that is the establishment of the cure collaboratories, that doesn't have NIAID's fingerprints, and in fact doesn't have Carl's fingerprints in some way. As Carl is going to describe over the next little while in a presentation, every 7 years the clinical research networks are considered for funding, and that 7-year cycle that the networks are currently under is nearing its end, and a new cycle about to begin, and Carl's going to describe that. Our plan for today is that over the next 15 minutes or so, Carl's going to present around the thinking at NIAID and the process of reaching future funding for the networks. Even as he's speaking, please do feel free to send questions either in the chat feature if you're on ReadyTalk, or you can email them to avac@avac.org, and we'll put them in the queue. And when Carl is done presenting, we will open the lines for anyone who wants to unmute, and we'll give instructions then on how to do that. And also I'll field the questions that will have come in electronically. I do want to say at the very outset that we really look forward to this call, not as a one-off that simply is a chance for Dr. Dieffenbach to present NIAID's approach and thinking. It really is not just the beginning, it's been an ongoing process, but this is part of a process, and we hope that this webinar will accelerate that process through which input is provided to NIAID as they think about the future. So that's the run of show. Again, delighted to welcome Carl Dieffenbach here to present on the thinking behind the clinical research network. So Carl, with that, I'll turn it over to you.
Thank you, Mitchell. It's a pleasure to join you. I'm waiting for the first slide to come up. There we go. So as Mitchell stated, this has been a dialogue that we've had ongoing since late January, and it's really important that AVAC host this opportunity for me to speak today, simply because we're seeking all avenues to get input into the science and the scientific agenda as we move forward. Next slide, please. And the most important issue is if you think about HIV and the speed with which we make progress, we really can consider the progress in 10-year cycles. So where we are today in 2017, when the next round of these clinical trials networks ends in 2027, what is the agenda that we wish to accomplish by 2027? It's a really important way of framing the conversation. You know, where are we today? Where do we want to be in 10 years? So as such, every 7 years we competitively review the work within the networks and then consider where the progress has been made, where we can refine and focus and refocus our efforts, but with the goal toward, by establishing this forward-looking agenda, we can really then have our form follow our function, and we can very aggressively then pursue a very specific and focused agenda. So on the lower part of the slide are 4 chevrons. The one outlined in red, the planning time, is where we are right now, and we'll talk about the others in a little bit. Next slide, please. So if you think about the progress we've made to date, there's been pretty spectacular work that's been done in prevention and treatment in terms of improving therapeutics, the development of PrEP, the development of sex-specific protection methodologies, whether it's with the ring through the ASPIRE protocol and the IPM protocol, or adult medical male circumcision. We've also performed the research that demonstrates that full implementation of option B+ can literally eliminate or virtually eliminate perinatal and breastfeeding transmission. It moves the questions in prevention of mother-to-child transmission out of the realm of research and into the implementation realm. We're building on RB144. We are also developing long-acting technologies. With this sort of as a background, next slide, we need to go back and consider what the NIH has specified through this post that the NIH director, Dr. Francis Collins, put out in August 12th of 2015. And this really helped us refocus and really made it so that, you know, the goal of the research should be, what can we do to reduce the incidence of HIV infection? If we're going to control the epidemic, we need to reduce incidence, and we need to then also develop the next generation of therapies that give improved safety and ease of use, which is part of treatment as prevention. We need to be able to discover and develop a cure for HIV. And ultimately, we also need to reduce the burden through prevention and treatment strategies for the HIV-associated infectious and non-infectious comorbidities. And all of this is underpinned by an outstanding programs in basic research, health disparities research, and we need to continue to train the next generation. Next slide, please. So as we look 10 years down the road, here is a straw dog for people to consider. Are these reasonable goals, or goals that we are seeking to establish for where we want to be in 10 years? Can we get to a point where we'll have long-acting, durable prevention and treatment strategies that are effective for at least 6 months? That would be an injectable or implantable or some other delivery system that would give us 6 months of protection or 6 months of treatment. HIV vaccines that have a greater than 60% efficacy, the ability to functionally cure a large set of HIV-positive people of all ages, normalize life expectancy for HIV-positive people, and ultimately also reduce burden of the HIV-associated infectious and non-infectious comorbidities, including TB co-infection. So with these as a set of goals, can we then take these next slide. What do we need to achieve these goals by 2027, and how should we refine the clinical trials networks moving forward today for the future? So if you think about it, what we're trying to do is anticipate the direction we are seeking to get to. So this is September, and it is fall now, and so we can start using hockey analogies again. So what we want to do is what the great one, number 99, Wayne Gretzky, always said he did, is he would skate to where the puck will be, and that made it easier for him to score as many goals as he did in his career. So what we're trying to do is anticipate where the field will need to be there with a research structure and infrastructure and support so that we can accomplish these kinds of goals. Next slide. So what can we do from the NIH perspective? We can continue to foster biomedical innovation. Additionally, through partnerships with the behavioral and social science groups here at the NIH, can we identify behavioral patterns and ways of intervening to improve adherence, and improve uptake, and improve testing, and also to understand what it takes to have a product be useful and desirable, particularly for adolescents. We want to continue to forge and nurture partnerships to extend our reach. But at the end of the day, if we focus on innovation, we also then want to be able to facilitate the partnerships with implementers so that they can come in and help us take whatever we develop to scale so that we can truly have a profound impact on the state of the epidemic. Next slide, please. So let's focus on the top two bullets. Next slide, please. So as we move forward, again, this is a process of these conversations that have begun back in late January. And as we've gone through, we've received a lot of feedback to date. And what it comes down to is we think that based on what we've heard to date, there are three key areas of research emphasis that will need to be developed, driven by interventions to cover all the appropriate target populations. And those are HIV prevention, HIV vaccines, and therapeutics research. Therefore, as we go forward, we're considering essentially a triad of clinical trials networks, one focused on HIV prevention, one focused on HIV vaccines, and one focused on therapeutics. And what I'd like to do over the next couple of minutes is walk you through the research agendas that we are currently considering in each of these three areas that have been built on the feedback we've received to date, starting with HIV prevention. Next slide, please. So we think about the HIV prevention scientific foci. There are three key areas. One is novel biomedical methods of HIV prevention. What are the behavioral and social science partnerships that will be needed to help achieve a full implementation and understanding of the biomedical prevention methods? And ultimately, the goal is to develop products that can protect the populations most at risk, specifically focused on adolescents, young adults, and U.S. minorities. This covers adolescents around the globe, such as young women in sub-Saharan Africa. Next slide, please. So what do we want from prevention tools? We need them to be safe. We need them to be safe first and foremost. They need to be acceptable. They need to be desirable. They need to elicit in people the desire to use the products. We can't have situations where people feel stigmatized by the prevention strategies we are offering them. They need to be highly effective at preventing HIV acquisition. And after long and careful thought, we want to make sure that the products that we bring forward in the future provide systemic protection. And an additional one that I think is one that is worth considering is is there a role for the next generation of post-exposure prophylaxis? This is an area that has not had any innovation since the early development of antiretroviral therapy. And groups like the City of New York are doing some truly amazing things with innovative delivery of PEP, but given our ability to improve the antivirals, are there innovative dosing formulations that also could be applied to PEP? But ultimately, we need to define the needs through behavioral and social science type research, the needs of the most vulnerable populations, tailor fit the prevention strategies where possible, and then take those interventions and focus on the populations most at risk. If we do not protect the youth of today, we will not have a future. it is absolutely essential that we focus on youth, that we focus on adolescents. By adolescents, I mean ideally in the range of 10 to 24. That's what our colleagues at Child Health use as a definition of adolescence. But ideally, we would be able to get to a point where we could have interventions that we could use well before the age of sexual debut. Next slide, please. Moving to the vaccine side of the house. Ultimately, where the vaccine world needs to live is demonstrate efficacy, demonstrate that they have a vaccine that is safe and effective. Then we can take it and use it to define the correlates of protection. And I have a separate question on this slide, is what are the criteria that we will need collectively as a vaccine research field and the community to help us focus this question, is what are the criteria that will be required to move a vaccine concept toward licensure. On the next slide, I spell out this in a little bit more detail. And so, next slide, what we clearly want to be able to do is once we have reasonably determined safety of a product and we have the support of the pharmaceutical sponsor, can we start initiating studies in adolescents and younger children of an HIV vaccine before efficacy is determined? We want to be able to determine the correlates of protection. We want to be able to bridge to phase 2 and phase 3 studies for licensure and market. And this is absolutely something that partnerships will be required. But another area that I think we need to emphasize is, should we be successful in developing a safe and effective HIV vaccine, we're going to have to work on improving the durability of that vaccine concept and also be able to streamline and simplify the vaccination schedule. All of these will be essential activities in a vaccine focused network between now and 2027. Switching to therapeutics, next slide please. Therapeutics has 4 areas of research: novel and durable therapies, cure, tuberculosis, and other complications and co-infections. So if we think about novel and durable therapy, here's the example of where the long-acting formulations, whether it's long-acting pills, go back a slide please. Please go back a slide. I'll keep talking what's on the words that just flew by while our colleagues at AVEC back up the slides. What we're seeking are novel and durable therapies that have long-acting formulations that are fully suppressive and are dosed infrequently. We need to also be able to continue to partner to take the newest antiretrovirals, get them licensed for use in pregnancy, at birth, through childhood and adolescence. In the cure space, we need to be able to establish virologic control in the absence of sustained therapy from infancy through adulthood, and that means working with immune-based interventions, immune activation as a driver of persistence, and also consider, where appropriate, gene and cell-based therapy. We absolutely need to have novel and durable therapy for better therapy for tuberculosis. We need to define new treatment and prevention strategies across the lifespan, including a TB vaccine. And we need to partner to license the newer TB medications during pregnancy, at birth, through childhood and adolescence. And ultimately, in adults, we need to really be able to focus on complications and other co-infections, including durable, defined curative strategies for hepatitis B in adults as we move forward. Now we're to the slide on leveraging partnerships. As we look forward, we want to continue to foster and nurture partnerships to extend our reach and facilitate the handoff. Next slide, please, which will discuss the partnerships that are required.
Carl, we seem to be having a bit of a frozen computer problem across a number of computers.
Okay.
Why don't
so you want me to just keep talking, Mitchell, so that we can get to the questions?
Absolutely. We're going to be posting the slides, so apologies to everyone, and particularly Carl, to present on a frozen deck, but we're working our best to unfreeze.
Okay, well, in order to keep us on time, I'll talk about partnerships, and then we'll talk about how the partnerships will be utilized for this for the next steps. So what we have, NIH research in HIV is really built on leveraging these essential partnerships, and there are partnerships within the NIH, within Health and Human Services, and across the federal government, like with PEPFAR. Additionally, we have partnerships with the private sector. We do not get innovative drugs or vaccine concepts without partnerships with the pharmaceutical industry, and ultimately through partnerships with foundations and with foundations and NGOs, can we then do this kind of testing? For example, all of you are aware of the strong partnerships between the NIH and the Bill and Melinda Gates Foundation. But ultimately, what we are seeking is when we prove the concept of whether it's a vaccine, a prevention, or a therapeutic, can we partner with implementers to facilitate the handoff, take a team approach, so we can then quickly figure out how best to implement a strategy and then be able to take it to scale, because ultimately that is what we are about, is changing the public health face of HIV. Can we get to a point where we are reducing incidence, we are building partnerships that will allow us to fully implement these novel strategies? So on the next slide, we have 4 buckets that talk about the types of partnerships that we'll need to continue to foster. Will be to develop combination prevention and treatment strategies, and then be able to evaluate them for how best to take them to scale. We need to be able to partner to prevent and treat infectious and non-infectious comorbidities, primarily focused in adults, improve the initiation and adherence to prevention and treatment, whether we're talking about a care continuum or a prevention continuum. There's a lot that needs to be done in this, and each location, each epidemic or component of an epidemic will have its own special challenges in terms of how to improve initiation and how to improve adherence. And ultimately, there are significant cross-cutting issues among these 3 leadership groups dealing with infants, children, adolescents, and young adults. Next slide, please. So this slide is not just a pretty picture. If you think of what HIV has done to communities before the time of therapy, it was essentially a forest fire where the trees were burnt down. And what we are seeking to demonstrate here is that through our research, we essentially have a new growth forest. We want to continue to foster the growth of the forest. So one day, we will essentially have eliminated HIV. We will have a case where essentially we're back to full aspirin regrowth. Next slide. So where are we in our process? So between now and January, we are continuing to foster feedback on the research agenda and other feedback that you may want to give. In January of 2018, I will be making a formal presentation to the AIDS Research Advisory Committee about the size, scope, and direction of our network plans. We will then start going into a quiet mode where we will write our funding opportunity announcements, publish them, hold the competitions and the application reviews in 2019, with awards in 2020, with then a continual discussion in the next award cycle about our future needs. And the last slide is we do want to hear from you. We need you to tell us what you think, whether you have a big vision or a specific note. But what I do need is time. And so if you could please get me your comments by November 30th, this will give me sufficient time to digest and process the suggestions and creative ideas that can come from you, the community, and the researchers that are listening today. So those are the addresses where you can send your notes. Additionally, if there are organizations out there that wish to have me make presentations or have discussions or dialogues specifically on aspects of this, you can contact me through the NIAID Network Refinement address there, and we will follow up and set something up with you as individuals. I appreciate the comments we've received to date. There have been some really outstanding ones, and we continue to take comments in and adjust based on the new and better ideas that we keep hearing. So Mitchell, thank you for hosting us today, and I look forward to questions that your audience can bring up.
Great, Carl. Thank you so much. That's a fantastic starting point and summary in terms of your own and the division's thinking about not just clinical trial networks, but about the HIV research endeavor overall, and enormously grateful. A lot of great questions that came in. Some very specific on specific questions that you have prompted. Some are kind of some cross-cutting issues, and then some larger process issues. And I'm going to try to group them together as best I can. To start, as you'll see on the screen for those on ReadyTalk, separate from the ReadyTalk chat feature, which you can continue to put questions in, you can email at avac@avac.org at any point. And after a few questions, I'll go ahead and open the line, and people can hit *7 to unmute your line. I will ask people to introduce themselves, be brief with the question, and then go back on mute *6 because we are recording this call. But incredibly good questions already coming in. Let me start with a very specific question, Carl. You said it a couple of times, both in the prevention presentation, or the prevention portion as well as the treatment portion, and this issue which we've heard so much more about in the last year or so about long-acting modalities, and a series of questions from different people, but one that really nicely summarized it. You kept referring to 6 months, or at least 6 months long-acting. What's the magic of the 6? Is that what you think is most likely based on existing research, or is that what you sense is the most optimal time point or the minimal time point? So just you could kind of talk both about the prevention and treatment space in terms of what's optimal when we think of long-acting.
So I tried to stress I would think at least 6 months. Ideally, I'd like to stretch this to a year. I think that it is possible to get to a 6-month window. And it depends on what the modality is, whether we're talking about a modified broad neutralizing antibody or a small molecule. I think that there's a reason to have it be in a situation where you could set it and forget it and then come back in 6 months. I think there's a destigmat from a behavioral and social science perspective. It's very destigmatizing to be able to take a shot or have some sort of delivery of a protection or a treatment. So then you don't have to worry about it for a period of time. And I think that this kind of a strategy, in combination with the continued work that the pharmaceutical industry is going to do on longer-acting pills, I just think we're trying to be innovative and push the envelope. But I think 6 months is in some ways a minimum. I think that the broad neutralizing antibodies will definitely fit within that. Drugs, we'll have to wait and see. Are you there, Mitchell?
I was there talking on mute to you, Carl.
Fantastic.
A very important thing. I was saying.
Okay, so I didn't hear the question. Maybe you could rephrase it
I'm going to
say it again.
I'm going to say it again because only I know what the question was. You had talked about PEP, which is something that doesn't get talked about, probably the least discussed in the research enterprise. And someone specifically wanted to know more about your mention of New York City, not only about PEP, but in your conversation about partnerships. The role of NIAID and the network's connections with municipalities, many cities and now states, with ending AIDS, getting to zero approaches. Where does the research, particularly with the 2027 vision, where does that interdigitate with these approaches from cities and states, and how are you partnering at an operational level, research to implementation in the sense of, say, a New York City or San Francisco?
That's a really important question. And I think it's really essential that we realize that not everything fits within the network model. And where we have been really quite successful in partnering with cities has been through the Centers for AIDS Research. And we have really used the Centers for AIDS Research because, again, what they are about is local control of a research program at a university or universities within the city. For example, in Washington, D.C., Alan Greenberg has a CFR that utilizes all of the existing universities within the jurisdiction. We just funded a CFR in New York that is highly leveraged and linked in with Dimitri and the city to get things going. The CFR in San Francisco is heavily involved within the public health infrastructure, and of course in Atlanta you have the ability of the Emory CFR to link directly to CDC, and through these kinds of partnerships, we've been able to work with the CDC to involve the cities directly and their health departments with the CFRs, where the CFRs can bring a research set of questions and help to build out those activities. So to my mind, there's a multidisciplinary set of tools that the NIAID and NIH have, and I'd say that when it comes to city activities, that's where the CFRs will probably play a more predominant role than the networks. That said, there are sites that are domestic that are the performance sites for our research clinical trials, and when those are in cities, the ability to attract and use those as leverage to attract attention to a set of research questions is a partnership that the CTUs or the clinical trials units can have to help foster the research and help to promote the work that's going on in their cities.
Great.
Kind of a complicated answer, but that's where we're at.
No, very helpful. And in fact, someone, funny enough, an advocate actually just wrote in and said, is it possible either on the website or do you have it available to send around, all of these different efforts, even within NIAID, so the CFRs related to the networks, related to the clinical trial sites, related to, say, the CDC. Is there some grand schematic that you have, much of what you just described, that would help people see the connections and what falls within these different categories?
Sure. So on the NIAID website, we have an extensive description of these activities that is there, and that's if you go to the NIH.gov, you can surf over to the NIAID website, or you can search it in Google directly and call it up. We have just recently reworked our website to make it more navigatable and easier to get around in. So all of this is available on the website.
Great. And although it is mid-afternoon on the East Coast, where you and I both are, Carl, I'm delighted that we have a number of African advocates and partners on the line. And on the call, and I think as we mentioned in the original posting, we do hope to have a second webinar with you geared to some of our international colleagues for whom it may be a little late right now. But a number of them are on the line still now and asking about outside of the U.S. So you mentioned the CDC and the HRSAs and obviously the cities and the CFRs that you just described. But can you help us understand what is the relationship outside of the U.S. and some people wondering what to ensure that this research and implementation is truly global?
Yeah, that's a real. So if you think about where the majority of the research that the NIAID supports, it's literally, I would say the balance is more international than domestic, although we've really tried to foster both. And where we need to continue to forge and nurture partnerships are in these partnerships that are international. PEPFAR, for example, has supported over the past several years 3 very large implementation science studies. One's called PopArt, one's called SEARCH, and the third was in Botswana. That really were looking at treatment as prevention. We need to be able to continue those kinds of partnerships for implementation. As we look forward on, assuming we have a safe, effective, and durable HIV vaccine, how are we going to partner with WHO and with foreign governments and with ministries of health to support the implementation of these kinds of activities? So on my slide, I actually had listed foreign governments as a major partner for us for the future, specifically to address this. And I think the researchers that are already in country will help us to continue to be the ambassadors to their governments, to help us know when the time is right to foster these kinds of handoffs.
Great. No, thanks, Carl. A question came in, and you mentioned it on the kind of research priorities in terms of, and one that I know you've thought a lot about and invested a lot in, and that's the CURE research agenda. And a question, just in terms of logistics, you recently funded the CURE Collaboratories, and a question about what's the interface between the CURE Collaboratories and the clinical trial network. Since you just funded those, a question came in, does that mean you won't be including those in the networks in this new era, or will you? So I think that if you think about where, so let's go back and realize again, like the CFRs, this is not a one-size-fits-all model. So what you need the collaboratories to do is to develop the translational research. And so can they come up with a schema or a methodology that may or may not have an impact, whether it's a therapeutic vaccine or some other strategy to either silence, eliminate, or control the HIV persistent reservoir. But ultimately, even if it goes through a collaboratory as a small phase 1, phase 0 clinical trial, we absolutely have to have a network structure to take it to the multi-center level. So within the therapeutics network, moving forward, is work on HIV cure across the lifespan. And I want to emphasize this, that there are activities that are essential for us to continue to do in pediatrics as well as in adults, and that will be housed within the network in a coordinated way. Because this is an example of where the field can synergize, because information we learn from adults will be directly applicable to children. And the advantage of perinatally infected children, even though each one is a failure on the part of prevention, is can we learn from each child to a point where we can essentially cure or prevent them from ever rebounding. And I actually think it may be easier to cure a child than an adult. And so I think it's really an important set of ideas and concepts that we absolutely have to have linked in the therapeutics network. But yes, there will be a handoff. It will be essential when we get to multi-center that these will be done within the therapeutics group. Great. That's super helpful, Carl. You know, a number of people commented how pleased they were about your mention of adolescents, and I think a lot of people commenting on, as you, I think, rightly point out, that if we don't, in both prevention and in treatment, deal with adolescents, we will never turn the tide on this epidemic. A question about the structures to support that. Obviously, there are adolescent issues across all of the scientific areas that you described. And so a question about how do you look at, say, a cross-cutting population issue, adolescents being just one, that would be obviously relevant to consider in the three you framed out: therapeutic, prevention, and vaccine. And is there some approach that would have a cross-cutting adolescent approach irrespective of what the research is? I'm putting together a number of questions that came in. I hope that I didn't.
Right. I understand the question. I understand the question. I think that's a really, actually a very insightful and far-reaching question, is if we had a situation where we had highly effective prevention that had been demonstrated to have high efficacy in adolescents, at the same time you had improved therapy, could there be this type of a cross-cutting implementation trial for how to reach and engage adolescents, use both a care and a prevention continuum model, but really engage adolescents in terms of being able to figure out how best to get them interested, engaged, involved, and adherent to the interventions. That would be a perfect example of that kind of a partnership that I would see moving forward.
Great. And actually, a similar set of questions, you mentioned very excitingly, I think, the social and behavioral science pieces. And there's never been a social science network or a behavioral science network. Obviously, it works in each of the networks. Someone asked, how is that being integrated so that, and maybe this is similar to the adolescent question, but how does the scientific agenda, say, around social and behavioral sciences get standardized and leveraged across the scientific endeavors in different networks?
That's a really good question. I think that's an area where we continue to seek input. As many of you are aware, we have formed a very close, mutually satisfying relationship with the National Institute of Mental Health, who's one of the leads for behavioral and social science research here at NIH. I think that as we go forward, it's going to depend on the question that we're seeking to address. So we are hopeful that we can continue to build on this relationship and continue to take the best and the brightest that these fields bring forward. And ultimately, the context of the behavioral and social science research may matter a tremendous amount. So we don't want to get into a one-size-fits-all situation. So what works for an adolescent young woman in the area around Durban, South Africa, may be completely different than a young black MSM, say, in rural Georgia.
So, no, thanks, Carl. I'm mute again. Really helpful. One other question, but just jumping back to adolescence just a bit. Someone asked about the challenge of what we consider, I guess, younger adolescents, the 10 to 16-year-olds. People, so much talk is about how do you get 16, 17-year-olds, given that much research starts at 18. But the 10 to 16-year-olds and a range of legal and clearly political issues for that population. So as you focus more on adolescents, is there particular thinking, and how will the networks and how will NIAID navigate some of those issues? That's
the $64,000 question, or in this case, $64 million question. I think that first and foremost is you have to have proven, innovative technologies or agents that have been shown to be safe and effective down to 18. And so step one is to have the studies really focus on being able to identify down to age 18, get those individuals enrolled, understand the adherence patterns of those individuals. And once you have safety in that population, then work with the host country governments, the ministries of health, so you have something that will work, so that we can start talking about implementation, and then work within each of the individual political structures to solve these issues. There's no one answer this question. So much of it deals with parents and behavior. It remains a major challenge. But what we've been able to do with therapy is get systems in place where we can rapidly move to younger and younger children who are HIV infected and get the drugs licensed and approved. Prevention is going to be a tougher challenge simply because of the heat and light around the mode of transmission. But there is no magic bullet at this time that we have to solve that issue other than continuing dialogue.
Thanks, Carl. I think it's safe to say there will never be a magic bullet for that one, but important to know the thinking behind it. There is a series of questions about numbers, and again, want to see if people are reading things into things. You presented with 3 scientific foci. You had a prevention foci, a vaccine foci, and a therapeutic foci. And question came up from someone saying, why are prevention and vaccine separate? And then another question related, I'll throw you at the same time, is seeing the way that you presented here, is one correct in assuming that there are likely to be 3 networks, a prevention network, a vaccine network, and a therapeutic network?
So I'll start with the first question first. And I actually wrote a blog post specifically around considering a linkage or a merging of the prevention and vaccine network. And this is an example of where you listen to people who offer cogent arguments. And there was a lot that was a lot of discussion around this topic. And what came out is that the tools and technologies needed to implement the research for early vaccine work and early prevention science work is so different that it just didn't make sense to try to merge it into a single network, but rather when we get to more complicated, bigger studies, to require those 2 networks to partner to address the cross-cutting issues that would be associated with implementation of large late-stage clinical trials. So more of a nuanced answer. It's not that we're creating independent groups, but we're creating groups that are interdependent on each other.
Go ahead,
on the question of number of leadership groups, as of today, the number of FOAs that we intend to publish would be 3 FOAs, 1 in prevention, 1 in vaccines, and 1 in therapeutics, and then a separate FOA for site activities. And I think there I want to stress the importance of the special populations and the target populations that will be such a key component that we will be seeking for the site activities. Because without quality sites that can recruit and retain the appropriate populations, all of the leadership group work is for naught.
Great. Super helpful, Carl. A bunch of questions came in about some of the products and modalities to be studied. And on the one hand, you talked a lot about the need to tailor and think about desirable products, what people really want, something we've all been talking about a lot over the last several years. And on the other hand, the focus on long-acting, and in the earlier question, looking at a 6-month or longer long-acting method. And I think a lot of people feeling we need that it's both and, not either or. But really wondering, with the shift to systemic, on what basis is that made? If we really are trying to understand the user and desirability of what they really want, on what basis do we think they will only want? And are we saying, or are you saying in this framework that people only will shift and the research will only shift toward long-acting systemic? Is there still room for user-dependent methods? We hear this a lot with adolescents particularly, who may not want a product in their bodies for many months or years at a time in their early sexual lives. So a question really about how do we kind of match this idea of understanding users and giving them what they want but saying we think they don't want user-controlled methods. And more specifically, I think a lot of people wondering where microbicides fit in the future in the framework that you've laid out.
So if we are talking user control, I think maybe let's go back and talk first and foremost about reality and making sure that we have to understand that the purpose of biomedical prevention is to protect an individual from acquiring HIV. And it's not like HIV is a benign pathogen. It is a deadly disease. And once you are infected, you're infected for life. So the prevention strategy needs to match the challenge that the pathogen is throwing at us. Additionally, there are possibilities for user-based control that also would give you systemic protection. I think that those need to be considered. But as long as sexual transmission occurs via multiple routes, you would want a product that was easy to use, that was able to be given once, and was less behaviorally dependent than current strategies. And I think that that's the challenge we face. As you talk about adolescents in one voice as not wanting something, but also probably the group that is least able to adhere to user-based control would be an adolescent. So I think we really need to be careful about what people want, and then is it pharmacologically possible to give people what they want? And it may not be. I mean, everybody would like, something simple, easy, the size of a microdot. I guess I'm showing my age. That would give you tremendous pleasure and satisfaction. It may not be possible. And so I think we need to think about what's reality-based strategies to give us effective prevention.
Great. Thanks, Carl. Related to that, one of the things that we've seen come out of a number of trials and much of implementation work with young women, and PEPFAR's talked about this a great deal in their DREAMS initiative, the intersection, of course, of contraception and HIV prevention, and a question about where, if anywhere, would the so-called multi-purpose products, a product with dual purpose, fit within the network structure? Is that something that would be prioritized?
If there was a multi-purpose prevention product that protected against all routes of transmission, that would be fine.
And is it something that would be considered within the prevention network as a top research priority? Again, this issue that I think...
I think it has to be considered within the context of what other... choices will have to be made, and we'll have to, given the nature of funding, you pursue the things you must pursue. What is the value added? We know that we have a ring that is potentially moving toward licensure. What is the next step for that ring? And the Microbicide Trials Network has a 300 milligram ring that is moving into evaluation. Does that provide systemic protection, or is there only vaginal protection still with that kind of a ring? Is it rational or reasonable to ask somebody to use a ring and a rectal product simultaneously? I mean, at some point it can become what's more complicated, remembering your daily use activities or a long-acting product that requires an injection once every 6 months. I think there's a lot of behavioral and social science work that needs to go into this. We've pursued rings, we've looked at gels, we've looked at pills, and where the innovation, we need innovation. And it just so happens that currently we have injectables and implantables, but there are other delivery systems that we cannot conceive of today that need to be developed and tried. This is where I'll continue to push for innovation and not necessarily get locked into, I want this, I want that, but let's free our minds to consider alternative innovation strategies.
No, innovation is clearly key, and I think this idea of looking, beyond the short term, and as I think the first time I've heard on an AVAC webinar, a Wayne Gretzky quote about going where the puck will be, not where it already is. Always a really important reminder. I do want to just get to any other questions on the line, but one of the questions that came up early on that I think quite apropos to the way you just finished this is with your own vision, that beautiful photograph you showed for 2027, is there a sense of a price tag? How much money do you think you would need to fully implement that vision? Something, again, I think for advocates who want to know, is that something to be pushing for? Is there a price tag that is in the context
that's
of these
a question that is not, I am not able to answer. I think the safest thing, I would like to be able to sustain the budget I currently have. I think that's about as far as I can go on that one.
Understood. I think for many of us, flat being the new normal and the new baseline is a harsh reminder of the world in which we currently live. I realize we're near the top of the hour, but I do want to just check. I know there are other questions that have come in that we will likely not be able to get to, but I do want to see if anyone does want to hit *7 and very quickly ask Carl a question directly. Again, I'll ask you to introduce yourself and be brief.
So this is Linda D. I have a quick question about cure research and immunologic non-responders, people who have really beat up, compromised immune systems. So we hope that they'll be prioritized too, and they're going to need immunomodulators and maybe vaccines or some combination thereof, as well as other treatments. So is that on your radar, Carl?
So Linda, thank you for the question. Going back to the earliest days of us talking about biologics for treating HIV, we've always had in the back of our minds this idea that there were cytokines and other chemokines that may need to be given to essentially enhance the regrowth of the human immune system, particularly after it's been beaten by HIV. So I think first we need to have some form of success and then refine it and improve it and make it more and more generally applicable. I see this as part of the research continuum. But ultimately, as I said, I would like by 2027 to be able to have sustained virologic remission within a large segment of HIV-positive people of all ages, and that would obviously include this population as well. So it's part of the vision.
Great. Thanks, Carl, and thanks, Linda D. Any other questions? *7 to unmute your line. I'll pause for just a minute.
So for those of you who would like to have your questions answered, this is the ideal opportunity to go back and look at the screen, write down the email addresses, paste them in, and send us your questions directly. And we will be happy to get you back answers to whether you have a specific note, a very focused question, or you have a big vision type question that you wish to address. So there's some selfish self-promotion on my part because I really feel that this is an opportunity for all of us to contribute together to make what we do here at NIH better through the input of AVAC, of the people who are on the call, and others that are interested.
Great, Carl. Thank you so much. And we did want to put those addresses on the slide that are on the screen now, and we will circulate those and put those in the posting following up the call. And a question actually related to all of that, Carl, in terms of process. Someone asked, will comments be publicly available as they are for some other federal government processes?
So that's I'm struggling with that one. And so when we started collecting the comments, we had not said to anybody that we would post their comments and questions. But what we are doing is going back to everybody who has written a comment and asking them how they feel about us putting it out either anonymously or with attribution. I feel kind of conflicted on this, that I do owe it to people to protect their privacy. At the same time, if they and I understand if they don't want to have their comments shared, we will. So my staff is busy reaching out to everybody who has written to us, and we have the idea that we will post a comment and an answer to those. And I just think it's the fair way to go. I don't know, Mitchell, if you have experience or have an opinion on that.
Well, I actually, you won't surprise you, Carl, if you have an opinion. I actually think to the degree possible, letting that be open would be terrific. I think we've seen that in other processes. I think it will spark healthy conversation within the community at large, which is clearly something you've committed to in this whole process. And so to the degree possible, obviously if people want to keep them confidential, that's their prerogative. But I'd love to see it. I think in the absence of that, one possibility would be, as you've done already a few times in the last several months of posting blogs, it may be that you could in a couple of blogs highlight issues that you really saw come up frequently, to give you a sense of what's on people's minds, or ones you absolutely were excited about and want to take on as true innovation, or perhaps one or two that you disagree with, but tell the community why. And again, those could be anonymous contributions, but ones that would understand the thinking. I think what we're hearing from advocates throughout the HIV Continuum, treatment, prevention, cure advocates in the U.S., in Europe, in Africa, is people wanting to be part of a process, and you clearly are setting it up to create a process. So to the degree that you can keep that going, I think all of us would be grateful. I think all of us would benefit from it in that spirit of really co-creating something, even though at the end of the day you are the funder and the researchers are the researchers.
I agree. We'll see what we can do. Additionally, I will have a new blog post that will probably be up within a week.
Great.
That'll
and
cover
I know
some of the same ground we just covered today.
That's great, and your staff has been super at getting them out, and we will be sure to pass those on to people here. And as we talked about, our hope is to have another call early in a day where many others in Africa and Europe can participate. If you have a couple more minutes, there are a few extra questions that we'd love to throw your way.
We can keep going a little bit.
Well, go ahead.
No, that's fine. Let's just keep going.
We have a lot of people on the line. They want to hear from you. One of the questions you had in your slide, rightly and somewhat provocatively, what will the criteria to move a vaccine concept forward? Here in the slide you had for licensure. I think a couple of questions came in about, given, as you said, the challenging resources that inevitably are going to be, choices are going to have to be made. Is the process by choosing not just candidates for licensure, but more importantly, candidates for next level of efficacy trials, is that something that would be part of the FOA and the expectation for the vaccine network to define, or will those criteria be developed in a separate process by NIAID and or with other funders and product developers?
That's a good question. I think the process of selecting candidate vaccines to enter efficacy testing has been a relatively large, complicated process that ultimately ends up with consultations at the NIH level with stakeholders. I think in past it's been part of the network to set criteria. And I think we'll continue to do that, as well as continue to take in input on what next from stakeholders. So an answer is all of the above. Okay.
And a question came in. And again, I think for a lot of people on the outside, it is a bit of an alphabet soup, and to figure out who's doing what. Early on, you presented a slide about the research priorities from the NIH director, Dr. Collins. And then you obviously focused a great deal, rightly, on NIAID and the Division of AIDS' work. And someone asked a question, how do the networks and the research agenda overall, is this NIAID's interpretation from the NIH and NIAID only? And how does it relate to the other institutes and particularly those that support networks? And how does all of, I think some people wanting to just get a sense of how does that all fit together?
That's an important question. Our single biggest NIH partner is the National Institutes, the Child Health Institute. They help support the work in pediatrics, on women, and adolescents. And I think that it is a highly valued collaboration that we are going to. We've met with, Dr. Fauci and I have met with their new institute director and will continue to do so to make sure that we foster the best work in that area. That said, we are subject matter experts in infectious disease. When it comes to the non-infectious comorbidities, we rely on other institutes to help lead that work. A prime example of that is the Reprieve trial, which is testing a statin to see how it impacts cardiovascular disease. So it's essential that everybody, each institute, play their key role and can continue to do so as partner. But ultimately, we have the Office of AIDS Research here at NIH that is in principle responsible for helping to coordinate. And that's a key role, is where possible, can they help build the bridges between NIAID and say, Heart, Lung, and Blood, or another institute to look at questions of aging, if there was an intervention that needed to be studied to impact aging. So I think that they've been essential partners in this and have played a big role in helping us get the Reprieve trial up and off the ground, for example.
Great. Speaking of infectious diseases, there was a question that came in earlier, actually from a colleague in Zimbabwe. So we have people up late tonight asking about sexually transmitted infections and where they fit into the HIV research agenda and for the networks, if at all.
Right. Well, that's an important question because there were, going back in ancient history to some studies that were done on prevention of, can you change the course of HIV acquisition by treating STIs, and there were a number of studies that were done on that. I think the treatment of STIs as part of dealing with the human subjects enrolled in HIV prevention studies is something that we've always taken very seriously. But research on sexually transmitted infections gets to a critical area that is actually housed in another part of NIAID. And we're working very closely with that component, with the Division of Microbiology and Infectious Diseases. So as they have products or materials that they would like to evaluate against an STI, could they essentially use our network structure to evaluate those kinds of activities in both HIV negative and HIV positive individuals? So that's an area that is an ongoing discussion with Division of Microbiology and Infectious Diseases. Many of you may realize that there's a new director in that division, and their new director is Emily Erbelding, who was the deputy director of the Division of AIDS. So we have a colleague and a friend there now, and are looking for ways to collaborate and synergize this type of research.
Great.
Again, that work would be done specifically with funding that doesn't detract from the AIDS dollars, nor would it detract from the ongoing HIV work at those sites. I realize there's some sensitivity with this. But ultimately, if we have this wonderful infrastructure, why wouldn't we use it to improve human health?
That's great and very exciting. I think we all look forward to you and Emily really forging even greater collaborations there. One other question, back to adolescents. Someone came in, and again, some people know, we have lots of people on the line, many of whom know the networks intimately, some of whom don't. Someone asked the question, where does the Adolescent Trials Network fit into this? And given that you're talking a lot about the adolescent focus across for 3 different areas, prevention, treatment, and vaccines. Does that mean there wouldn't be or need to be an adolescent trial network?
The Adolescent Trials Network, just for everybody's edification, is a child health supported set of activities that is focused primarily domestically and has sites around the country that deal with questions of adolescent HIV prevention. I would say that there's opportunity for collaboration between the ATN and what we are doing. The ATN is not really set up to do interventional trials that require INDs and that level of scientific requirements. They're just not. The institute isn't set up that way. So partnership there will be essential in both ways. They have sites and activities that the NIH-funded networks could support and foster. So it would be a great type of collaboration. And in fact, there are ATN sites that are part of the cabotegravir trials that are ongoing at this point, as just an example. So yes, that's an ongoing collaboration already.
Great. Thanks so much, Carl. One, I know we're now quite over, but one more question that I think is really important for, I think, all of us, but particularly for those of us at AVAC. Working across a lot of different research areas and across all the different networks, we see quite different approaches to community engagement and stakeholder engagement. And again, as you look forward to 2027 and a slightly different architecture of the networks, where might community engagement, what might it look like? What should it look like from NIAID's perspective? And I guess that's true for both the networks, which lead many of the research protocols and design, but also at the site level where the real work obviously happens at the community level. So what's NIAID's thinking? Is that will it be part of the FOA? Have you thought about that? I think a lot of us on the line have, and wonder what you're thinking.
Yeah, I think that's a really important question, and it is a question that continues to confound simply because how much is enough? And the answer is probably there's never enough, but what's a sufficiency that'll get the work done in an ethical, appropriate way? So this is one I struggle with personally all the time. So actually feedback on this from the community would be quite useful. We have systems in place that deal with cross-network activities, whether it's HANC or other activities associated with our network structures. But again, they serve a very specific focused purpose of dealing with community engagement around specific protocols, around specific interventions. Do we need to be broader than that? And if so, how much? And realize that it's a zero-sum game. For every $1,000 spent on that kind of activity is probably $1,000 that doesn't get spent directly on an intervention. So there's a balance here that we need to agree is appropriate. So feedback on this point would be very useful.
Great. Well, that is great to hear. As it happens, and you are likely aware, we recently at AVAC, with a number of partners, hosted a global think tank on good participatory practices. We were delighted that a number of the NIAID-supported networks were there, as was HANC, and a report from that is forthcoming, and I think is a great baseline perhaps for some of that conversation going forward. So we will definitely follow up on that. You've been incredibly kind and gracious with your time, Carl. As people will see up on the screen again, there is both the email address, NIAID Network Refinement at mail.nih.gov, and the NIAID.gov HIV Research Enterprise link for more information. Carl, before we do finally close, any last thoughts, comments, or questions for this audience?
No, I just want to thank everybody who stuck with us for the past 75 minutes to hear this conversation. I look hearing from you, whether it's a big vision or a specific note. And Mitchell, we look forward to following up with you to get a follow-up webinar scheduled.
Fantastic. Well, thank you and your team for putting this together and collaborating with us. And we are really excited with the opportunity for input. And I will just say, not that I can speak on behalf of everybody on the line, but knowing that you've spent a lot of time with the clinical trial networks that exist already and with the research community, your interest and desire to have input from those outside of the research structures as they are normally conceived is a terrific start, and we all look forward to contributing to the conversation, and lots of follow-up, and we can certainly talk about, in addition to the extra webinar, also looking at ways to help create this conversation through the comments that you've already been receiving and how that all plays out in the future. So just a great thanks to you. I want to thank everybody for joining in, particularly those of you joining in from late at night. I've seen some emails from people, and I'm grateful that you stuck with us this late, and we look forward to many more opportunities, because at the end of the day, it really doesn't matter if it's NIH funded or pharmaceutically funded or philanthropically funded. This is an HIV research endeavor writ large, and we all, to call your major point in your presentation about partnerships, recognize that we've all got to engage in this in all of our different ways. So really thankful to you for being part of this conversation, and look forward to many more opportunities.
Thank you, Mitchell.
Cheers all, and have great afternoons or evenings. Cheers.